Quality tolerance limits, or QTLs, are predefined boundaries around a small number of critical, trial-level parameters. They are designed to flag when trends suggest the reliability of the trial or the safety of participants may be at risk. The point is not to monitor everything equally, it is to focus oversight on what is truly critical to quality. QTLs sit within risk-based quality management, which current good clinical practice, or GCP, guidance expects teams to apply. Instead of treating every metric as equally important, sponsors identify the data and processes that matter most for answering the study question and protecting participants, then put targeted controls around them. QTLs provide one of those controls at an aggregate level. They help distinguish a systemic issue from a local glitch, for example, when patterns emerge in discontinuations, protocol deviations, or missing primary endpoint data. Designing QTLs starts with the study risk assessment. Look at the objectives, endpoints, design, and population, then ask which failures would most undermine interpretability or safety. That typically leads to candidate parameters such as completeness of primary endpoint data, the rate of major protocol deviations, or withdrawals for safety reasons. Each QTL then needs a precise definition, including how it will be calculated and which data source it relies on. Setting the limits is part evidence and part judgment. Teams usually draw on historical studies, feasibility work, and clinical insight to understand what is expected, then agree the point at which the integrity of the trial could reasonably be questioned. The key is to avoid treating QTLs as a standard template. They should be study-specific, clearly documented with a rationale, and integrated into the quality management plan rather than added late as an afterthought. Operationally, QTLs work best when they are embedded into existing oversight tools and routines, such as dashboards in a clinical trial management system, electronic data capture, or a business intelligence layer. Parameters should refresh often enough to spot trends early and remain visible to both sponsor and vendor teams. If a QTL approaches or exceeds its limit, it should trigger a defined process. That usually includes reviewing by site, region, and time, looking for patterns, performing root cause analysis, and then agreeing whether the signal reflects a data artifact, a local operational issue, or a broader systemic problem. Conclusions and any corrective and preventive action, often called CAPA, should be documented to maintain a clear audit trail. It also helps to separate QTLs from other measures. QTLs are reserved for parameters where crossing the limit could affect overall trial validity or safety. Key risk indicators, or KRIs, are often site-level signals used to spot local outliers. Key performance indicators, or KPIs, track efficiency and delivery, such as timeliness. Good governance makes QTLs usable. Cross-functional input from clinical operations, data management, biostatistics, pharmacovigilance, and quality helps keep QTLs lean and relevant, with clear escalation rules and regular review. Common pitfalls include having too many QTLs, never revisiting them when the risk profile changes, or poor communication that leaves sites and monitors unsure what a breach means. When QTLs are designed and governed well, they act as an early warning system that supports safer decisions, stronger data quality, and fewer late-stage surprises. To learn how Quanticate can support your next trial, email inquiries@quanticate.com or request a consultation through our website. Thanks for watching, and don't forget to subscribe for more practical insights on clinical trials and modern drug development.